G. If I don't have any question, I will proceed with the next next technique of targeted genome editing, which is called as the TALEN. So in the earlier class, we talked about the zinc finger nucleases, and I told you that the zinc finger nucleases were considered as prestigious at that time because they able to clip, they able to mutate, they able to delete a place on the genome by designing appropriate zinc fingers in array. These are also called as zinc finger arrays, and in general they're called zinc finger nucleases. The complications with the zinc finger nucleases is that one zinc finger, please have an eye on that thing. Let me find that that one zinc finger, this one zinc finger is able to detect or attach itself with 3 base pairs. One zinc finger is able to attach with 3 zinc fingers. This is a limitation. Second thing, every 3 base pair traditionally should start from the G. So this is the left side, sorry. This is traditionally it should be a G rich region. This is another type of limitation, you can say. So you need to find an appropriate sequence using different combinations of zinc fingers, and most of the time all the zinc finger combinations available will not be able to create all possible sequences, because one zinc finger is actually recognizing 3 base pairs. So sometimes one of the base pair in the sequence is missed because of this limitation. And also the off-site activity of the zinc fingers nucleases was very high, and the efficiency is very low. About 2 to 3% efficiency was there. It's nothing, it's very laborious. But scientists were very happy at that time since they're able to do it. Remember, in research, the first step is to have an experiment done, is to have a result. No matter how much money should be, or how much money will be consumed to have that experiment done. The first thing is to get things done, to show somebody that it is possible. The next and follow-up work is to reduce the cost. In order to understand this thing more easily, imagine the Human Genome Project. Most countries present in the world, along with different big giants, pharmaceutical companies and other biotech companies, they collaborated to sequence the total human genome present in human cells. It's like a drive executed by full resources of the planet. It consumed 10 years. It requires 10 years, and every country or every research institution divided their work, like chromosome number 17, fragment number this, sub-fragment number this belongs to me. Chromosome number 12, segment number this, sub-fragment number this, sub-fragment number this belongs to somebody else. So everybody sequenced those components separately, and then we put them together to make human genome. It took 10 years and millions of dollars to find that thing. Now, today, we can have that thing done in a day with only $1,000 or less than $1,000. So 10 years versus 1 day, millions of dollars, $1,000. for the sequence of one genome. So the first thing is to have things. So people were so happy at that time that they able to target a one particular place on the genome. So the next thing is to improve that technology. So I was doing my MS at that time. I was such a bad student at that time in South Korea. When that paper rocked the world, the paper came from the people in Germany, and they have discovered a pathogen, a bacteria, while they were studying the pathogenic properties of that bacteria with respect to rice. So it was nothing to do with mammals, humans, and mammalian cells. They were working for different reasons. That bacteria was so smart that when that bacteria infect the rice cells, it not only infects the rice cells, but they diffuse or silence the specific cells present in the genome of the rice, which are responsible for the immune system, which are responsible for the defense mechanism of the rice. So what that means, when bacteria managed to enter into the rice cells became defenseless because the bacteria somehow selectively switch off the defense genes. The scientists were very amazed at that time. They investigated that how that bacteria is able to go to a specific target places, how they selectively silence and block one particular gene present in the rice genome. They ended up with a protein which they called as TALs or TAL protein. They called it as a TAL, transcription activator, like uski tarah ki protein. It's a very small protein. When they studied and characterized that protein, they came to know that this one protein can recognize one base pair on the rice genome. So one protein is able to recognize one base pair. There are plenty of TAL proteins, many of them able to recognize the 4 base pairs very easily. So we need 4 TAL proteins only, 4 TAL proteins which should be able to recognize A, C, G, and T. And the next thing is that we just need to put them together in the order of our own choice. We need to put the TAL proteins in order of our own choice to target what? To target the place of our own choice. The rest of the story is same. It is associated with the fork protein once again. Can you see? Associated with the fork 1 nucleus. Another TAL array associated with another fork 1. When these two TAL proteins successfully finds their places, this one and this one, the fork 1 dimerizes and then create a double stranded break at the place of your choice. The principle was same, but what is the most interesting thing here, that one TAL protein is targeting one particular base pair. That means we can target any sequence. All we need to do is to put the 4 TALs in a sequence of our own choice, and we will be able to target anything. If you put 12 TALs together, you will be able to target 12 base pair, and that is good enough specificity. So the complications, limitations associated with zinc fingers are now gone, because 1-to-1 base pair correspondence is available. When scientists studied the TAL proteins further, they came to know it is just because of the 2 base pair, just because of the 2 amino acids. It is just because of the 2 amino acids present in the TAL proteins the two amino acids present in the tall proteins, which allows the tall protein to capture a base pair. For example, an Ng. So all the structure of the tall protein is same. Structure means amino acid sequence. All the amino acid sequence of the tall protein is same except two amino acids. If those two amino acids are Ng, the tall will going to bind with the T. If those two amino acid sequence will be HD, the tall protein will going to bind with the C. If those two amino acids will be Ni, the tall protein will going to bind with the A on the DNA. If those two amino acids will be NN, the tall protein will going to bind either with the G or with the A. So by putting these four different types of tall proteins in the order of your choice, you will be able to generate a DNA binding protein of your own choice. And then all you need to do is to put the fork one. The sequence is cloned in the bacteria. For example, here, can you see here? This plasmid will going to generate, this is the DNA sequence which will going to produce the tall array. And then you can see fork one here, and this fork one will going to be part of the talls as well. When this plasmid enter into the cells, it will produce the tall array with the fork one, and then those DNA binding protein will go and attach themselves with the place of your own choice, eventually leading to the dimerization. That means eventually leading to the double stranded DNA break, and the rest you know. So that enhances the capacity of the scientist at that time to target theoretically every single base pair, every single place on the genome. So that while from their work, from the pathogens, they generated that idea to engineer tall proteins together with the fork one, and it is called as TALENs or tall effector nucleases. If you compare them with the zinc finger nucleases, you can guess that it should be more easier to engineer TALENs. It will be more easier and more specific as well. And it founds to be much better than the zinc finger nucleases. But still, the efficiency is too low that it is not something which you like to have in research and in medical sciences, because it is associated with lots of laborious work. So the scientists still working to improve that technology, but at that time they ended up into entirely new mode, and that what we know as CRISPR-Cas9 system. It is entirely different from these machines. What you have learned is about creating a DNA binding domain. In case of zinc fingers, you create a DNA binding domain of your own choice, bind it with a nucleus. In case of tall effectors, you create a DNA binding domain of your own choice. So in both cases, you are creating a DNA binding proteins, associate them with a nucleus to create a double stranded DNA break at the place of your own choice. Now the principle will be changed in the upcoming techniques. So this story is done here. All you need to know, what are zinc fingers, their specificity, and about the tall effectors, what are the difference between these two. It's very important question. Just keep this thing in your mind for any GRE and other things, or from any other professor who is working on these techniques. So let me summarize the path which life sciences were taking at that time. In all those years, actually in all those centuries, at least since the dawn of life sciences, I must say. So there were certain people who were setting the principle of biology, and you can name them. Darwin was one of them. He designed like a theory which is acting like a Qaradat-e-Maqasid, as if Qaradat-e-Maqasid in Pakistani constitution. You know, Qaradat-e-Maqasid is not in Pakistani constitution. It is like a document which is superior to the Pakistani constitution. So these laws are like covering everything, controlling everything. You remember the Mendel and the principle of inheritance, and then the people who discovered and came to know about what is the material, who is the material, who is responsible for the transferring and the transmission of information from one cell to another, and that was nucleic acid. They're like the principal founder of everything. And then we have people who have generated the information about that material. We know MacLeod and McCarty. We can remember and respect, if we want to, Watson and Crick. But please do not forget the beautiful Rosalind Franklin. And then the genetic code was discovered. And then we came to know how to manipulate that genetic material, how to deal with the genetic material. We came to know restriction sites, restriction enzyme. We came to know how to sequence it. We came to know how to amplify it. And then we sequence the whole genome. We came to know everything about that genetic material. And then the ultimate wish which we have, how to edit that genetic material in a way we want. We develop zinc fingers, we develop talons, and then we are working on CRISPR. This is amazing discovery. This one enhances the capacity, and this is like everything for us about targeted genome editing. So if you see that slide in a nutshell, a weird thing you can sense, that like everything is working for that one purpose. And you know what is that one purpose? That one purpose is to edit the genome so that we can create a life of our own choice. This is the ultimate wish of life. This is the ultimate wish which we can have, that we should have a life form which should look in a way we want, we should act in a way we want, we should be able to expand our capacity, we should be able to extend our lives. Everything is actually running towards that one thing, and genome editing is a bottleneck. Genome editing is a bottleneck. Once we have that capacity, once we have that ability to manipulate the genome successfully with more than 99.99% accuracy, we will be in a different world altogether. After that, there is no going back, I am telling you. After that, the one who have the resources will be the best. The theory of natural selection will be changed. The concept of life sciences which we know will be changed, because we will be able to implant abilities to life after that. And that's what we are working on. Who knows that we will be able to reach it, or we might, destroy our planet before that time. So that's